Hi, I'm Kyle. I'm a PhD student at Stanford University in computer science, and I'll be talking about our work at UniTox. And so what we're interested here is really trying to build a data set of drug toxicity. So for instance, one thing that we're really interested in is trying to understand if we could predict drug toxicity ahead of time so that if a drug is going through clinical trials, we can know beforehand whether it will be toxic or will not be toxic. There's some existing data sets already that have some of this drug toxicity information, but the problem is that actually curating this sort of toxicity information is a very manual process. So for instance, if you wanted to build a data set of drugs with cardiotoxic information, FDA scientists would actually manually read through FDA drug labels, read through tens or hundreds of pages of these drug labels, and try to decide if a drug counts as cardiotoxic or not cardiotoxic. This is really not a very scalable procedure, so we were really interested in seeing if we could apply large language models to automate this process so we can more rapidly decide which drugs are toxic and which drugs are non-toxic. So here we're introducing that dataset. It's called UniTox. It's a large multi-organ toxicity dataset curated by large language models. So first I'll explain the pipeline. So our pipeline looks something like this. We start with the whole universe of FDA-approved drugs, and we look at all their drug labels, and we first focus down to the most relevant drugs, about a set of 2,400 FDA-approved drugs. For each of those drugs, we then take its full drug label, which can be anywhere between tens and hundreds of pages, with all the clinical trial information, all the adverse side effects of that drug. We'll then feed that label into GPT-4o, and we'll ask it to summarize that drug, and specifically summarize the toxic effects of that drug. With that summary in hand, we'll then go back to GPT-4o again and ask it to give us a rating of how toxic that drug is, either a binary rating of yes, no toxic, or a ternary rating of somewhat toxic, very toxic, or non-toxic. Once we have those ratings, we now have our data set, and then the question is, how well did our model work? And we do two different evaluations, one on external pre-existing data sets, and one using some clinician friends to help us decide if our model was accurate or not. So just to show you what our data set looks like, we have 8 different toxicity types. For each of those 2,400 drugs, we have a distribution of non-toxic, somewhat toxic, and most toxic drugs. This is what our drug structure, or data set structure looks like. We have each drug, the different toxicity type, the label for how toxic it is, as well as a summary that was produced by GPT-4o explaining why it's toxic or non-toxic. First, we compare on some external databases. We look at cardiotoxicity, liver toxicity, and renal toxicity. We compare our model's predictions to the previous manual gold standard labels. And we see that our predictions are actually very accurate. And what's interesting is that even for the cases where maybe our model makes a mistake, it looks like it's different from the pre-existing manual label, often our model was actually correct. So if you look at the summary, it actually used updated information and is actually more accurate than the pre-existing label. So this is, if anything, maybe a lower bound on accuracy. We also tried ablating a few different versions of the model, so changing the prompt a little bit to use keywords, removing the summary, or using a different auto type, and found that our original model structure was probably the best and the simplest, and also gave us these nice summaries for extra interpretability. And then outside of those 3 existing databases, we also tried going to some clinicians for our other 5 toxicity types, and we asked them to tell us for each of our models predictions of drug toxicity based on their clinical knowledge, was our model accurate? And we found that our model had actually a lot of concordance with what clinicians said. So this is how we built our data set and evaluated it. And then the last part was actually looking at how we could use this data set for some cool downstream analyses. So first thing we did was look specifically at small molecule toxicity, and we wanted to ask if we could build models that would look at the structure of a drug, and just from that structure of a drug, try to predict the toxicity of that molecule. And so we trained a variety of different machine learning models. We tried some graph neural networks, some random forest methods, and basically showed that across these different toxicity types, we had actually pretty reasonable accuracy training these models on human in vivo toxicity. And definitely another thing that's really only enabled by this sort of large-scale multi-organ toxicity dataset is the ability to do cross-toxicity analyses, where we could look at different toxicity types for the same drug and see how often the same toxicity type appears for that same drug. And then finally, what we were really interested in was seeing how much risk the FDA is willing to tolerate for toxicity in drugs. So for instance, there might be certain drug classes like anti-cancer drugs where the FDA is much more willing to accept some toxicity because it's really important to take that drug in order to save the life of the patient. In contrast, there might be other drug types like maybe anti-symptom drugs or anti-pain drugs, where the FDA is much less willing to accept toxicity. And so we're able to see that by basically analyzing the toxicity predictions of our model and then looking at how those toxicities change from one drug type to the next. And so that's our UniTox data set.